Okay, great. So for this session, we'll shift the focus from cosmology to biology. And our first speaker is Luca Giomi, who is a theoretical biophysicist at Leiden University. So Luca, the floor is yours.
Can you hear the microphone? Now you can, I guess. So good morning, everybody. Indeed, as it was introduced, so this is going to be like a smooth transition from the previous session to this session, because the talk is about, indeed, the information universe, but in biology, and specifically in the context of bacterial colonies. Now, being a father of two little children, I often find myself thinking and talking about bacteria. Germs, microbes, whatever you want to call them, right? For obvious reason. But because I'm also the husband of cosmology, I keep hearing about Hubble expansion, Hubble tension, dark energy, modified gravity, all those nice topics that I'm sure you know everything about. And on top of that, I'm a theoretical physicist myself, and you know how theoretical physicists are. They like to unify things, right? So I've been thinking for a few years, is there anything common between microbiology and cosmology? Now, bacteria are known in the Netherlands since a long time, 1673, when Anthony van Leeuwenhoek in Delft, after looking inside his home-built microscope, first realized that there is an entire universe at length scales below 1 millimeter. Back then, it was not yet the information universe, but I promise you that by the end of this talk, it will be. Now, bacteria are extremely successful and resilient form of life on Earth, possibly the most resilient form of life on Earth, because they're able to colonize nearly any surface, even when the environmental conditions are extremely primitive, for instance, in terms of temperature, humidity, and so on. As they do so, they can form some very beautiful patterns, like in the work of Ben Jacob, who's one of the pioneer of bacteria physics in a way. But can also produce diseases, of course, and diseases whose severity ranges from a very simple tooth cavity to a life-threatening cystic fibrosis. Okay, so it's very important to understand why bacterial colony forms and how they are organized. This is a little expansion history of a typical bacterial colony. So this was done in particular for a type of bacteria called B. subtilis, but you can think of it as a proxy for all kinds of bacterial colonies. And cells are initially swimming in a bulk fluid, okay? And then eventually when they find a surface, they attach to this surface, and then something very dramatic happens because they undergo what is called a phenotypical switch. They get rid of their flagellum because they don't need it anymore, they don't need to swim anymore, and then they start to secrete an extracellular matrix, namely some kind of mucus made of proteins and sugar that they use to attach to the surface, and eventually they start to divide, okay? And as they divide, they first form a monolayer, so like a single layer of cell that keeps expanding in space. And eventually there is a second transition, but this time not a biological one, a physical one. And this monolayer becomes a multi-layer structure. So, a structure that features multiple layers. This multi-layer structure grows, okay, goes into contact with the bulk fluid again until some of the cells switch back to the initial swimming phenotypes. This is called palantonic phenotype in biology. And eventually leave, take off, go back swimming in the bulk fluid, and possibly they start another colony. So with my group, we have focused on the first two stages of this transition, okay, of this process, which are closer somehow to the mindset of a physicist. And the last paper that we wrote, that will appear next week or the week after on Nature Physics, deals with this transition in particular, and demonstrates this transition is actually extremely robust. Namely, it happens always at the same time with respect to the age of the colony. And I think this is something very interesting. The way I like to think about it is that it's a little bit like puberty. Puberty is like an important transition in our life. Now imagine if the onset of puberty was like uniformly distributed across your lifetime. It would be a disaster, right? Instead, puberty happens exactly when it's supposed to happen, and so this is this transition. okay, and many other things in biology, but this time we know why. Instead, today, I want to tell you about this part of the process when these bacteria are still in a monolayer form. So this is how it goes. See, this is a video that was given to me by my friend Anupan Singh Gupta. What you see is E. coli on a substrate. So they grow, they form a colony that consists of some 500, 600 cells. And then eventually you see this dark, this white region in the middle. That's precisely what the cells are actually being extruded from the monolayer to the multilayer. Okay, so this is what you see here is precisely the onset of this transition. Now, I don't know if, if the cosmology in the audience will look at that as something that is analogous to maybe the Hubble expansion or to an expanding universe and expanding space and so on. But, for us, the first time we saw that video, the first thing it took, it came into my mind was instead another system in soft matter called a liquid crystal, and more precisely, what is called an active nematic liquid crystal. So I will explain these words in a second. So first of all, forget the word active. A nematic liquid crystal is a fluid which is anisotropic, and because it's anisotropic, this gives the fluid very interesting optical properties that, for instance, you can appreciate if you sandwich the fluid in a cross polarizer. Now, if you now imagine to zoom on a little volume element of this fluid, what you will see is molecules which are elongated and roughly aligned along the same direction. Now, these molecules are polar. They have a polarity. For instance, this red ball here, which is supposed to represent a nitrogen atom, could be on the right or could be on the left. And yet, when they pack together, especially at room temperature, all that matters is the shape, not the polarity. Okay, and as a consequence, they align along a common direction. So we can think about them as rods, as if they were rigid rods aligned in the same direction. And this average direction is usually called with the letter N, and it's called the nematic director in the language of soft matter physics. Now, unlike a rod, a vector does have a direction, okay? So if you construct a theory of nematic liquid crystals, this theory better be invariant with respect to rotation of the nematic director of 180 degrees, okay? So mapping n to minus n should not change anything in the theory. And there are various ways to implement this symmetry. The most elegant one that was introduced by Dejean consists of introducing a so-called order parameter tensor, the tensor Q, which is proportional to the tensor product of this vector n with itself minus its trace. And S here is a scalar number in between 0 and 1, which expresses how much the orientation of this rod deviates from the average. So it's something that you can think of it as the variance of this distribution. This quantity is called indeed the nematic order parameter tensor and is manifestly invariant for the rotation that I told you about. When n goes into minus n, because here we have a product, the object Q does not transform. So any theory that is revolves, that is expressed in these variables will also have the right symmetry. Now, this is a nematic liquid crystal, is what is inside your iPhone or your computer screen. An active nematic liquid crystal is a nematic liquid crystal whose building blocks, in this case bacteria, are able to autonomously move or generate mechanical work. Okay, for instance, by dividing, or for instance, by swimming. So in this case, what happens is that you have a very similar situation to the one that you had before, except that now the motion, okay, of these bacteria will result into the fact that the boundary of this volume element experiences a force, a surface force, or as we call it, a stress in mechanics, which is of course proportional to this direction n times a constant that somehow embodies all the biochemistry of the process. So the forces exerted by these bacteria, the velocity, the rate of at which they consume chemical energy, ATP, and so on, is all somehow embedded inside this constant. So this is a very simple theory. In mechanics, also, people typically don't like to use this stress vector, possibly because of the ambiguity of the sign. So it's much more common to use a stress tensor from which the stress vector can be computed. Then if you do this exercise, if you construct the stress tensor corresponding to the stress vector, this happens to be precisely proportional to the Q tensor, to the nematic order parameter through the same constant that we introduced before. Okay. Now, the reason I'm showing you this, I hope, simple equation is because, in a way, they illustrate the magic of active nematics and, in general, of active matter. Okay. This is because this Q tensor is an order parameter, so it's a quantity that becomes non-zero when a symmetry is broken, right? But it's also a stress. It's also a physical stress. So if you think about a passive material, take a ferromagnet, when there is a phase transition, when a paramagnet becomes a ferromagnet, the way the part of the system interact with one another does not change. And in fact, we use the same Hamiltonian for both phases, right? Ising, Heisenberg, you name it. This does not happen in active matter. So in active matter, in active nematics in particular, when there is this phase transition, that comes with a whole zoology of forces, stresses, internal interactions, which generate mechanical work. And because we are talking about biology, you can bet your house that the system will actually use these internal forces to do something useful, because that's how life works. So that's an example of a numerical simulation representing the expansion of a bacterial colony. So this is a discrete simulation. We simulate cell by cell. And as they expand in the plane, the color indicates the orientation. What you can see is that there is indeed a lot of alignment inside this colony, but for some reason, this alignment is sort of confined inside domains. Okay, it does not propagate uniformly throughout this colony. Okay, but it's confined in these domains. And when we saw this for the first time, this simulation was done by my former PhD student, Young Yu. Well, we wanted to say really something about the geometry of these domains, which is a stochastic geometry. Of course, they still can be characterized. For instance, you can say, what is the average area of this domain, or even better, the probability distribution of this area. Because biology is a bit easier than cosmology when it comes to experiments, we also went to our friends and asked to, try to repeat this type of data analysis with real cells and tell us whether we were heading towards something real or just a property of the model. And he came back with this plot. So these are two different bacterial colonies. This is E. coli. The cells are different for the aspect ratio, so the ratio between the length and the width. And as you can see, these domains do exist. And their area and shape and distribution also seems to depend on the aspect ratio. For instance, in the case of longer cells, okay, you can see the domains are much bigger. We wanted to understand that, but in order to do it, we had to introduce also a criterion to actually identify these domains. So this is exactly a result of numerical simulations. As you can see, we can reproduce the existence of the domains, and we can also reproduce the fact that somehow their shape, their distribution changes with the aspect ratio. If you look at the orientation instead of the domains, you will see that there is no preferential orientation inside this colony. So the isotropy of space is somehow reflected in the overall distribution of the orientation of all the domains. So as I told you, we introduced a bit of a criterion to decide which group of cells represent a domain. This criterion is actually not very important, but we defined a domain as a connected cluster of cells whose orientation differs at most by 3%, which corresponds to delta theta equal 0.1 radians. Okay, and this will give, for instance, these as a domain disconnected from the others. If you change this threshold, nothing happens. So the quantities change, the magnitude of the various quantity changes a little bit, but not dramatically. All the scaling properties remain the same, unless you really make an extreme choice. For instance, if this is 100%, then all the cells are in the same domain, but that's clearly not a good criterion. So here is a few data about this domain geometry. So this, I'm sorry, there is a curve that here does not appear, it's blue. This is a probability distribution function of the area of these domains, okay? And as you can see, this probability distribution is exponential. So except for this little region, which corresponds to the cells that are at the boundary of this colony, so they are loosely packed, And in that case, also, a domain is not properly identified, except for this region. This is a linear log plot. You see a line, so this is an exponential distribution. Similarly, the area of these domains, okay, does not seem to depend much on their position, okay, but only on the doesn't seem to depend much on the position. And instead, the area grows with the aspect ratio and decreases with the growth rate. So I'm a bit late, I'm sorry. These are experimental data that somehow confirm this prediction, even though they are not super good data. I will skip that and instead telling you why there is an exponential distribution and why there is a precise length scale here. Because an exponential means that there is an intrinsic length scale in the problem. This length scale originates from a competition between two torques. On the one hand, there is an active torque that attempt to disorder the cells, so rotate the cells, but on the other hand, this rotation causes entropy, and so there are passive forces which are trying to fight back, okay? And the two compromise at a given distance. You can think of it a little bit as a tug of war between two, people pulling the same rope, and this is the length scale that we see in this numerical simulation, in these experiments. This is the last thing I promise. This is a length scale, but why is it exponential? Okay. Well, in order to figure this out, we can try to do like this exercise and imagine that the colony is actually an ensemble of domains, and each domain can be characterized by two numbers: A, the area, and the number of domains having that area, okay, Ni and Ai. There are also two quantities that somehow defines a macroscopic state, and those are the total number of domains and the total area of these domains. And because the domains are indistinguishable to one another, you can count the number of domains that corresponds to these two macroscopic quantities equal to this multinomial factor. If you now approximate that for large n values, and you take a continued limit, you will see that this number actually is equal to the exponential of this function. And what is this function? That's a question for you, actually. Exactly. Is the Gibbs-Shannon information entropy. Okay, so it's really the information content of the system. If you maximize that, what you will find is the Boltzmann distribution, as usual, from which you can recover the exponential distribution. Good news for two reasons. One is that this is, to me, is one of the most simple, complex form of matter on Earth, like living matter, and yet we found that actually obeys, in a way, to the same laws of the simplest form of matter, namely a perfect gas. But more importantly than that, we did understood that indeed, von Loveneck, what observed in his microscope was indeed the information universe. Okay, because this is the distribution that maximizes the amount of information in the system. Sorry for going over time. Thank you for your attention.
Thank you so much, Luca. It's really interesting work. So I think we have time for one question from the audience. Yes?
Thank you, Luca, for a great presentation. I was just wondering, you leave out all the biological, biochemical dynamics. There are things like quorum sensing, there are all kinds of gradients there. And it's quite remarkable that then still you can recover those patterns. What would happen if I would just include these things? Would that then destroy these patterns again? Or so what is actually driving this? Is it really the physics only, or would it be something in combination?
Up to this stage is only the physics, really. I mean, so the biochemistry is there, of course, but it results in two things. One is that the cell grows, the cell divides. Those are very complicated biological processes, but physically speaking, in the end, is something we can make sense of. And the attachment with the surface. That's very important also to develop the type of friction that the cells actually need to go up. Okay, so this extrusion process is really like the competition between these internal stresses and friction. So at this stage of the development, this description is fine all the way to this transition. What happens afterward, though, when the colony grows, becomes a three-dimensional structure, then instead it rely much more on the biochemistry, because there are differences clearly between what's inside the colony and what's outside. there are phenotypical variation, and you see from the fact that eventually the cells are able to switch back to the swimming phenotype and take off. So we think that for this early stage, this is like an okay description.
Okay, thank you so much.
Yeah.
Sadly, we have to continue.